Welcome to our Unit 3 lab presentation, Structural Biology Analysis using PyMol. For our abstract, we said that by understanding each component of a protein structure, we can understand the recognition and cleavage function of the Cas9 protein. Our results support the hypothesis that the recognition and cleavage function of the Cas9 protein is related to the protein and DNA structure. Cas9 is a protein that can be used to alter a DNA sequence. It does this by recognizing a target area and using nuclease to cut the DNA out. There are some crucial sequence characteristics such as affinity and pH that can determine the structure, such as the grooves. The major and minor grooves of DNA in specific protein domains of proteins are shown to influence the protein-DNA interactions. For our introduction, we said that DNA is the genetic material in cells. There are three major forms of double-stranded DNA connected by interactions between complementary base pairs: A form, B form, and Z form DNA. B form DNA is a right-handed double helix that we are used to seeing. We use PyMol in these labs, a visual protein system, to look at molecules in depth. The central dogma tells us that DNA is transcribed into mRNA, which is then translated into proteins by ribosomes. In the mRNA, these amino acids are encoded as codons, or triple codes, that each specify an amino acid. More than 95% of protein sequence is from DNA sequence data. Molecular weight and the isometric point are two important properties for a given protein. They can be calculated from the amino acid sequence of the protein and can be used in protein purification. Each protein folds into a unique three-dimensional structure to perform its function. A mutation is a change in a DNA sequence. Mutations generate the variation on which natural selection acts on. Mutations can result from DNA copying mistakes, exposure to radiation, chemicals, or an infection by a virus. Alpha helixes are made up from the hydrogen bonds of carbon and oxygen atoms and the nitrogen and hydrogen bonds of the DNA. Beta sheets are made from three or more hydrogen bonds connecting two sheets in a protein. Protein-DNA interactions are fundamental to almost all biological processes. Proteins are recognized and bind to DNA in two different manners: sequence and non-sequence specific manners. Cas9 is a protein that can delete, add, and alter DNA sequences. It does this by recognizing the site of the change using cRNA. Then it swaps DNA sequences out by cleaving the phosphodiester bond in the DNA using a nucleus protein.
Hello, I would like to present our results and figures for our presentation today. We did 4 different labs that kind of took a deeper look into Project 1 Cas9 for the Cas9 system. So for lab 1, figure 1.1, we identified that the average distance of the hydrogen bonds in base pair was 2.9 angstroms. Interestingly, the adenine and thymine created 2 hydrogen bonds, where cytosine and guanine created 3 hydrogen bonds. And as you can see kind of here, this photo shows an adenine and thymine nucleotide base pair with the 2 hydrogen bonds here. For figure 1.2, we identified that the gray backbone has the 3 end, while the red has the 5 end. And we can really tell the difference because the 5 end here is connected to the 5th carbon, while the gray does not have a phosphate group sticking out. For figure 1.3, we found the distance between the nucleotide strand for the inner stack distance, and the average was 3.5 angstrom. And all 3 of these figures kind of show the important idea of affinity of the nucleotides. This is really important in identifying which is the major and minor grooves. And it is huge in determining the Cas9 protein interaction. Here for lab 2, we kind of dissected the sequence even further and found different characteristics of it. So the first thing is such as the number of amino acids, which is 4,107, molecular weight, PL, and atomic composition. In this lab, we identified that the pH really affects protein interaction, especially for this sequence. Any change in pH in the environment will disrupt the hydrogen bonds and can potentially change the shape of the protein, which will heavily affect its function. For lab 3, we identified that there are 13 atoms that are within the hydrogen bond here. And 3.2, this is the alpha helix shown, and it is a right-handed alpha helix. Alpha helix are right-handed, and the beta strands here, we identify that they are parallel. For figure 3.3, this is interesting as it shows the hydrophobic effect. So we see that the green is showing the hydrophilic while the red is showing the hydrophobic. And you can kind of tell because the green is more prevalent on the outside while the red is more in the inside. And that's because the hydrophobic portions of the sequence is I'm trying to avoid water, and so the hydrophilic side does a lot better job with interacting with water, so it's more towards the outside. For lab 4, we found that there are 20 nucleotide base pairs in this structure, and we found that when mutated, it would not be able to base pair with cRNA, which heavily affects, again, the function of the protein. For 4.2, this is a photo of the active nuclease site that can carry out the function of cleaving the phosphodiester bonds of DNA and RNA. So yeah, thank you very much.
Hello, I will be sharing the conclusion and discussion portion of our experiment. Our results support our hypothesis that the recognition and cleavage function of the Cas9 protein is related to the structure of both DNA and protein molecules. We determined that the double helix structure of DNA, and more specifically the major and minor groups resulting from it, are important to the DNA protein interactions. The major group participates in sequence-dependent interaction, meaning proteins are able to interact with the nitrogen bases in the DNA molecule. Through sequence-dependent interactions, the cRNA is able to locate and bind to the protospacer adjacent modifier, also known as PAM, so that the Cas9 protein knows where to cut the target DNA sequence. In lab 4, we identified this region of interaction on the protein-DNA complex. We were also able to identify the sequence-independent interactions along the minor groove, which occur via electrostatic interaction with the charged phosphate in the DNA backbone. These interactions are not sequence-specific, allowing Cas9 to interact with multiple different DNA strands. This is also supported by the uniformity of hydrogen bonds and interstacking distances in the DNA molecule. Because of this, the DNA molecule has a consistent shape, and the Cas9 is able to bind to any DNA molecule without changing its shape as long as the PAM sequence is present. The component of protein structure that is most relevant to protein-DNA interactions is the specific domain, which is a defined group that may contain the repeating structural patterns such as alpha helix or beta sheet. These specific domains function separately, so a mutation to the Cas9 protein outside of the functioning domain will not affect the function of the Cas9 protein as a whole. Overall, our results support our hypothesis and what we have learned about structural biology and CRISPR throughout this class.